And I was going to briefly mention, without overwhelming you, a few key themes here. I already began the story with genomic alterations. Genome means a complete suite of genes in a given cell or the cancer cell. And these alterations, how they start cancer and how they lead to cancer progression, how the immune therapy really goes after these things. And now we try to target the immune cells over time. And I just want to go back to early 2000s. And this was a monumental project done by two different teams. One team was funded by the US taxpayers, the NIH, the other was a private team. And they both, at that point, had mapped out the human genome completely. At that time, this was like a man going to the moon type of mission. Nobody had mapped out all the genes in the human cells. And this was a monumental 10-year project. And this project, really was very expensive, mapping one cancer gene, one normal genome. By the way, this is a normal, healthy, pooled sample from healthy individuals. And they mapped out what we call one human genome. It took 10 years, four institutions, $3 billion, B as in boy, for each of these projects. So for mapping one complete genome cost $3 billion, B as in boy, and 10 years. And this was done at, I'm proud to say, at Washington University, a building just behind me. And one-fourth of the work, one-fourth was done in Boston at the Broad Institute at MIT and Harvard. And one-fourth was done in Baylor in Houston, and one-fourth was done in the UK at Sanger Institute. And this was announced by the US president then at that time, Bill Clinton. This was a monumental, remarkable project that really gave us the framework how to study the genes in large numbers simultaneously and rapidly. So fast forward a few years down the line, this is the first instance where two of my colleagues, Rick Wilson here and Tim Lay, they mapped and published a paper in 2008. They mapped a cancer genome completely. This is one patient. The interesting thing is that they did two genomes, one from the cancer leukemia cells, one from the normal skin tissue. And they did this within a couple of years and for just a mere $1.5 million. This is the first instance ever a cancer genome was completely mapped out in all the 20,000 genes. And this was accomplished significantly for a very low amount of dollars. Remember, I told you the human genome mapping cost us $3 billion. This could be done. By the way, this is two genomes. We could get it for $1.5 million. And this is only in 2008, not long ago. And now this is a paper again published from my colleagues at Washington University. And this is published a few years ago, where the whole genome mapping was done within 5 days. And the very first human genome project took us 10 years. The leukemia project took us a couple of years or so. This one, now we can do this within 5 days. And the cost is around $1,000. So it has come down dramatically. That has really opened up our field for research and the use in the clinic. By the way, in the clinic, we don't do the whole genome mapping. We take about 400 or 500 genes that are really important for cancer, and that's what we map. So it gets done very quickly, and it's a little less, onerous for the processing. And so we really are doing it in the clinic. Right after the cancer genome was sequenced, around that time, the US National Cancer Institute started a monumental project that was one of the most impactful projects in projects in cancer. And this is called the TCGA, the Cancer Genome Atlas. Essentially, you're creating an atlas of the cancer genome. And this included 22 different types of cancers. And we mapped all the important parts of the genome completely. And over 11,000 patients participated in that, and 22 common types and 10 rare cancer types for a total of 33 different tumor types. And 11,000 patients participated in it. And we learned a lot from this project for various cancers, including lung cancer, breast cancer. And this really opened up a field. And now there are a number of projects that have gone on. This really helped us understand how genes get altered, how many genes get altered per tumor, what are the differences between smokers and non-smokers. and what are what we call co-mutations. You may have EGFR mutation, but something else could also be there that may play an important role in in how the cancers behave in a given patient. So we've learned a lot, and this really started that project over time. And I was very involved in the Cancer Genome Project right from the beginning. I co-led the Cancer Genome Atlas project with my colleague, Dr. Matthew Meyerson from Dana-Farber and Broad Institute. And this is one of our own work we did at Washington University. This is a very pretty picture. This is a patient's tumor where we mapped out all the 20,000 genes. It's a little hard to follow, but I'm going to walk you through that. It's not that hard to follow. You remember I told you the long stretches of genes are called chromosomes. So that's what you see here. And these are chromosomes numbered, 1, 2, 3, 4, all the way up to 22. And you can see that here each segment is a chromosome, and these all the letters that you cannot read are all those mutations or the letter change that I talked about. And these are, the squiggly line should be very straightforward here. These are number of copies of a gene. In this instance, there were some areas where there are too many copies here, too few copies. And so you can see a lot of copies here in this area. You can see a lot of these genes are extra copied, extra copies of the genes. And these lines represent a piece of the chromosome from here has gone and stuck to this chromosome. In this instance, it is a chromosome 7 going to chromosome 14. So this kind of a crisscross lines tells you how these things are jumbled. So it's fair to say that if you look at this patient's tumor cell, the genes are messed up in various different ways. Lots of these mutations, lots of these places where they're no longer two copies, they're either one copy or they are 5 copies or 10 copies or 15 copies, and chromosomes are all jumbled here, one part going here, another part going there. So it's really a messy, what we call messy genome. Then I'll show you, this is called Circo's plot, where we map out the entire genome. Then this is again, the next patient is again a patient of mine, lung adenocarcinoma. You see, this is very neat, very clean. You don't see the complexities you see there. You see very few gene alterations here. The copy is pretty even, more or less. And there is not much here. You can see those alterations here. There's absolutely nothing really going from one chromosome to another chromosome. It's pretty pristine. So if you look at them side by side, both are lung cancer patients. Both have lung adenocarcinoma. What's the difference? The difference is here. This patient was a lifelong smoker, got lung cancer. This is a non-smoker. So when you smoke for a long time, or when you smoke each time, I should say, you inhale 60,000 cancer-causing chemicals. They mess up the genes. And so those mutations accumulate over time. Not all of them play a role in cancer. Some do, but not all of them do. And so it's a pretty messy, when you map out these genes, there are too many mutations. And if you're non-smokers, there are few critical genes get altered, but that sets the stage for cancer. And so there are two different diseases in a way. They overlap some, but there are two different diseases. Does it mean these patients do poorly? Not always. There are patients, like this patient, for example, these patients have a lot of mutations, there are a lot of abnormal proteins, and this immune cells probably has been fighting these for a long time and get exhausted. This is where if that exhaustion is produced by the PD-L1 pathway, giving drugs like pembrolizumab or Keytruda or similar other drugs would make a big difference. And some of these patients do exceptionally well too. In fact, this patient of mine is still alive. And the non-smokers, on the other hand, have other issues that are favorable for them. They may not respond well to immune therapy, but they do have higher levels of these targetable alterations. So much so that in a paper we published a few years ago, we showed that 70% of non-smokers have some genes we can go after. We can target them with specific therapies. Maybe that's about the 10-15% in the smokers' lung cancer. Most common, of course, in smokers' lung cancer is the KRAS mutation. And this is the subject of our paper. We were asking the question in the TCGA, the Cancer Genome Atlas project, We took about 500 patients with lung adenocarcinoma and asked what proportion of these patients have genes that we can go after. And we found that we have about 75% potentially we could go after. But remember, a significant chunk, about half of them nearly, had KRAS mutations. Until recently, we didn't have anything for them. And even here, we have drugs only for a portion of this KRAS, portion of the slice. But this portion we can target. EGFR, a number of them we can target, BRAF, MET exon 14, and all the fusion alterations, and many of them we can alter, including the HER2 amplification. And but in about 25%, we can really find out what's driving these cancers. And we did several papers subsequently and then studies, and we really found out, we whittled this down to perhaps about 10% unknown right now. And this is a more recent paper. We found multiple explanations of why these cancers are growing, what genes are driving them. And so we now, with all this mapping, maybe I would say 80, 85%, we could find genes that are driving these cancers. In others, I think many times the good genes are gone, or there are multiple small things eventually have a cumulative effect in driving these cells to grow more and more. And so this is a work that we are doing currently to figure this all out. But a good number of these alterations are now targetable, more so in the non-smokers compared to smokers. And I also told you that the non-smokers and smokers are quite different in the amount of mutations they have. But if you look at this may be a complicated graph, but I'll tell you what is here on this side of the line is more common in non-smokers. This side is more common in smokers, and that's kind of highlighted here. EGFR, more common in non-smokers. KRAS, more common in smokers. And that's the bottom line. So there are differences in the way these things work out, even though they look the same.